Hey everyone, welcome back to another session of Biology and Human Welfare. In today's class, we are going to study about the Plasmodium vivax. Let's begin our class. Let's start with systematic position. Phylum is Protozoa. Subphylum is Epicomplex or Sporozoa. What is Sporozoa? In its life cycle, at least once there will be the formation of spores. So we call it as Sporozoa. Class is Telospore. Genus is Plasmodium and species is your vivax. It is the most common parasite in men. Do you remember in previous class we studied the most common enteric parasite? Write it in the comment box which one was it. Now it is digenetic. What is the meaning of digenetic? It is going to complete its life cycle in two hosts. Who are the hosts? One is your human and second one is your mosquito. Which mosquito I am talking about? It is female Anopheles. Female Anopheles. Now human is the secondary host over here and mosquito is the primary host. Whom we call it as primary host where sexual reproduction takes place. Secondary host, asexual reproduction takes place, right? Now in human they are going to infect cells intracellular way. What is the meaning of intracellular? Intra means within the cell. So it is going to enter inside the cell and harm the cell. Which cell I am talking about? It will be liver cell and it will be RBC, right? Now when we talk about the mosquito, it is going to infect extracellular. What is the meaning of extracellular? It is not going to enter inside the cell. Okay, it is not going to enter inside the cell. It will affect in between the cell. For example, stomach wall of mosquito. Stomach wall of mosquito, right? Now, this is the neat question class. The first cell which sporozoite is going to infect or the malaria or Plasmodium vivax infect, that is your liver cell. It is not blood or it is not RBC, it is the liver cell. Now, we already discussed primary host, secondary host. Reservoir over here is monkey. Why? Class, tell me why monkey is not being infected. Plasmodium vivax तो उसमें भी है तो infect करना चाहिए, right? Why it is not infecting? Because no development occur over here. No development of Plasmodium vivax occur over here. Now, what is the infective stage? Infective stage is the stage at which the parasite is going to enter inside the host body. So here it will enter inside the host body and it will start infecting the host cell. So what is the infective stage of malaria? It is the sporozoite, which is of sickle shape. यहाँ पे आप diagram देख सकते हैं. It is of sickle shape. Nutritive stage is nothing but at the stage at which it is going to feed. Feed करेगा तो grow करेगा. Grow करेगा तो it is going to affect the host. Which stage? Trophozoite stage. So infective stage is sporozoite and nutritive stage is trophozoite. Mode of infection is inoculation. Now, what is inoculation class? When you introduce small amount of things into something, we call it as inoculation. For example, अगर आपको दही बनानी है, if you want to make curd, how will you make? You are supposed to take a spoonful of curd and put it in the lukewarm milk. ऐसा तो नहीं करते कि हम पूरी की पूरी दही उठा के डाल देते हैं, ऐसा तो नहीं होता, right? We are putting a small quantity of curd. This small quantity of curd we call it as inoculum. Okay, the process we call it as inoculation. So what is inoculation? When you put small quantity of something and allow it to grow, we call it as inoculation. So यहाँ पे क्या हो रहा है? Small amount of sporozoite is going to enter inside your body and it is allowed to grow inside the body, infect yourself, right? Now, malarial parasite, if we'll see, 4 types of malarial parasites are there. Plasmodium vivax, Plasmodium falciparum, Plasmodium ovale, Plasmodium malaria. Before moving ahead, you have to be clear with the word tertian and quaternion. Tertian means third, quaternion means fourth. इतना तो हमें पता है. But what is third? What is this fourth? After every third day, you will have fever. That we call it as tertian. After every fourth day, you have fever. That we call it as quaternion. Now, another term which you should be aware about it, that is your benign and malignant. See class, benign and malignant आपने cancer में भी सुना होगा. When it stays only at the particular place, it is not spread all over the body, हम उसको बोलते हैं benign. So since it is staying at one particular place, what has happened? Is it severe? No, it is not going to be severe. Not severe. Not severe. इतना ज़्यादा harm नहीं करेगा आपको. But when we talk about the malignant, malignant में क्या होता है? Once area or one part of the organ or cell, anything is going to be destroyed. From there it moves to the other part, then it moves to the other part, then it moves to the other part. By this, whole body is infected. So tell me, is it going to be severe or not? It is going to be severe. So malignant is severe, benign is not severe. I hope you understood the meaning of tertian, quaternion, benign and malignant. So Plasmodium vivax is benign tertian malaria, which is the most commonly and widely distributed. That's why we are going to study about the Plasmodium vivax. Malignant tertian, malignant है ना, so it is going to be most dangerous. Mild tertian, rare in India. Quaternion malaria, that is less common, right? Now, the term malaria is taken from the Italian language which means bad air. Malaria का मतलब bad air नहीं हुआ है. Malaria जिस language से लिया गया उसका मतलब होता है bad air. World Mosquito Day is celebrated on 20th August as Sir Ronald Rose identified the oocyst. अब ये oocyst क्या होता है? You will understand in the life cycle. In the stomach wall of female Anopheles in Sikandrabad. 25th April is the World Malaria Day by WHO. Write it in the comment box what is the full form of WHO from 2007. Now, the whole scientist, whoever is involved in this Plasmodium vivax, I have written on this one page. I want all of you to copy it like this, okay? So, Charles Leverian, who was a French military doctor, discovered the Plasmodium in blood RBC of the malarial patient. Patrick mentions Scottish doctor while working on the elephantiasis. Now, put question mark over here. You will understand this. Malaria might be spread by the mosquito. Sir Ronald Ross, British Army doctor, awarded the Nobel Prize because he identified the oocyst of Plasmodium in the stomach wall of female Anopheles mosquito. G.B. Grassi and others described the life cycle of Plasmodium in the female Anopheles mosquito. Graham studied the ultrastructure of Sporozoites of Plasmodium. Schwartz and Graham discovered the hepatic zygogony. You won't understand, put question mark here. Camellia Golgi described the erythrocytic cycle, and Bano nucleus of the oocysts first undergo meiosis, then mitosis. Again, oocysts pe question mark. You will understand everything when we are done with the life cycle. Now, structure of sporozoite. It is the infective stage. Just now we have seen. अगर उसका ultra structure देखेंगे, तो this was given by Graham. Now, shape is nothing but the sickle shape. Length is 15 micron. Width is 1 micron. Covered by elastic pellicle. हमने pellicle के बारे में पहले भी पढ़ा हुआ था, right? Now, cell organelle. Okay, it is having microtubules also. Okay. Now, cell organelles, Golgi complex, endoplasmic reticulum, mitochondria, and nucleus. All these are present in the sporozoite. Convoluted tubule. Class, convoluted PCT and DCT. आपने 10th standard में you must have studied in nephron, nephron. Okay. It is going to be like this. इस तरीके से होता है. We call it as convoluted tubule, right? It is going to be many, where it is present in cytoplasm, present throughout the length of the body, and the function is unknown. Then it has a apical cap. इस तरीके से होता है class. इस तरीके से होगा. Now, if you see, there is a depression. Here, there is a depression. So, it has a cup-like depression. Present कहाँ पे होता है? Anterior side पर होता है. And it has a pair of secretory. इधर से pair of secretory organelles निकलते हैं. When I say secretory organelles, that means for sure it is going to secrete something. What is that? It is nothing but the cytolytic enzymes. Cytolytic means what it is going to break? It is going to break cell. It is going to break cell, right? Now, see here the structure. Follow the structure which is given in your textbook. So, this is your apical cup, and then this is your secretory. किधर लिखा? Pair of secretory organelles. Then you have nucleus. Then convoluted tubule. See this pink one. All over the body it is distributed. Then you have mitochondria, cytoplasm, pellicle is the outer body, and then microtubules spread all over the body as per your NCERT. Your Telugu Academy book. Okay. So draw like that. नाउ लाइफ साइकिल ऑफ प्लाज्मोडियम एंड वेन, तो हम लाइफ साइकिल ऑफ प्लाज्मोडियम वाइवेक्स हम दो वे में पढ़ेंगे, एक तो हम पढ़ेंगे मेन में, दूसरा हम पढ़ेंगे मस्किटो में. Before moving ahead, you are supposed to understand three terms. Life cycle of the Plasmodium has three stages. What are the three stages? It is sizogony, gametogony and sporogony. अगर हम sizogony की बात करें तो it is nothing but the multiple fission. We have already studied multiple fission is the asexual reproduction. Multiple fission is nothing but the asexual reproduction. So this multiple fission, when this occur in Plasmodium vivax, उसको हमने special नाम दिया हुआ है. We have given special name. We call it as sizogony. Now it is completely going to occur in human. It is of two types. One is hepatic sizogony and second one is erythrocytic sizogony. What is sizogony class? Multiple fission of Plasmodium vivax. We call it as sizogony. Hepatic sizogony is nothing but hepatic मतलब क्या होता है? Liver cell. So where it will occur? It will occur the multiple fission of Plasmodium vivax. When it occurs in liver cell, we call it as hepatic sizogony. Then what is erythrocytic sizogony? When multiple fission of Plasmodium vivax occur in RBC, we call it as erythrocytic sizogony, right? Now here see, second stage is your gametogony. Gametogony means formation of gamete. It is going to occur both in human as well as in mosquito. It starts in human but ends in mosquito. Sporogony means formation of spore. It is going to completely occur in mosquito. So sizogony completely occur in human, sporogony completely occur in mosquito, and gametogony it will occur in both, starts in human and ends in mosquito, right? So sporogony की अगर हम बात करें तो it will occur in mosquito where in the stomach wall of mosquito, right? Now अगर हम we are studying Plasmodium vivax in human, तो यहाँ पे हम दो stage की बात करेंगे, that is your sizogony and gametogony. Sizogony में we'll study about hepatic sizogony, erythrocytic sizogony, and gametogony में we'll study about the formation of gametes in human. So ये भी आपको टेबल बनानी है क्लास. I have sorted all the things in one table. Hepatic sizogony and erythrocytic sizogony. Hepatic sizogony was discovered by Short and Graham, and this was discovered by Camillo Golgi. Hence we call it as Golgi cycle. What are the phases which are involved in the sizogony? तो अगर हम hepatic sizogony की बात करें तो pre-erythrocytic cycle and exoerythrocytic cycle. वही हम erythrocytic sizogony की बात करें तो it has erythrocytic cycle. Now what is the infective stage? हमने overall infective stage स्पोरोजोइट पढ़ा था because it initiate, but अगर हम cycle wise पढ़ेंगे तो प्री एरिथ्रोसाइटिक साइकिल में you will be having sporozoite as an infective stage, क्रिप्टोजोइट एज एन इन्फेक्टिव स्टेज. अभी नहीं समझ आएगा, जस्ट सुन लो. ओके, नाउ ईदर क्रिप्टोजोइट ऑफ प्री एरिथ्रोसाइटिक साइकिल और माइक्रोमेटा क्रिप्टोजोइट ऑफ एक्सो एरिथ्रोसाइटिक साइकिल. कुछ भी नहीं समझ आएगा. वंस वी आर डन विद द लाइफ साइकिल, कम बैक टू दिस टेबल, यू विल अंडरस्टैंड एवरीथिंग. ड्यूरेशन इज ऑफ 8 days, 2 days and 2 days. Now resultant stage, infective stage हमने देख लिया, ये है. इन्फेक्ट करने के बाद जो बनेगा what it will be, cryptozoites form. यहाँ पे आप देख सकते हो cryptozoites infective stage. This is also known as first generation merozoite. And कितना cryptozoite? 12,000 cryptozoite. Then here metacryptozoites are formed, which is also called as second generation merozoite, and this is smaller and larger. Smaller one को हम बोलते हैं micrometacryptozoite, larger one को हम बोलते हैं macrometacryptozoite. And then erythrocytic sizogony में there will be the formation of erythrocytic merozoite, which is only 12 to 24. Class, don't get confused between pre-erythrocytic, exoerythrocytic and erythrocytic. See, pre मतलब तो क्या होता है? पहले होता है. That means it will occur before destroying the RBC. Erythrocytic means when it is destroying the RBC. Then what is exoerythrocytic? Is it going to happen after erythrocytic cycle? Not like that. See, same time infective stage is going to infect the fresh liver cell and it is going to infect the RBC. But the cycle in RBC is slower as compared to the liver cell. क्यों? क्योंकि यहाँ पे infective resultant कितना है? 12 to 24 है. But here you see 12,000. So which one will be faster? Liver one will be faster, right? So हमने इसके लिए लिखा हुआ exoerythrocytic cycle. That's it. Now ये सेम चीजें आपके टेक्स्ट बुक में लिखी हुई हैं. You can just read it once. Let's start with the hepatic sizogony. So mosquito will come. Mosquito इधर आ गया. Mosquito and mosquito ने bite किया. Okay. Mosquito comes and bite human. Once it bites, it is going to insert 2000 sporozoid. एक मस्कीटो आया, मस्कीटो ने ह्यूमन को बाइट करा, बाइट करते ही क्या हुआ, उसके सलाइवा के थ्रू 2000 sporozoites are going to enter into your body. Within half an hour, इस होने के बाद के within half an hour they reaches to hepatocytes. What are hepatocytes? These are nothing but your liver cells. These are nothing but your liver cells. They undergo two phases. Pre-erythrocytic cycle, exo-erythrocytic cycle. Repeating again, एक मस्कीटो आया, मस्कीटो ने आपको बाइट करा, बाइट करने के बाद 2000 स्पोरोजोइट अंदर गया. जैसे ही वो ब्लड के अंदर गया, माइंड इट, वो ब्लड के अंदर गया है, ब्लड में गया है, ब्लड को इन्फेक्ट नहीं कर रहा है. ठीक है. नाउ अच्छा, ब्लड जब सक करता है मस्कीटो, तो वी ऑलरेडी नो कि व्हेन ब्लड कम्स आउट इट इज गोइंग टू क्लॉट. बट मस्कीटो में क्यों नहीं क्लॉट होता है? क्योंकि उसका सलाइवा में एंटीकोआगुलेंट इज प्रेजेंट. एंटीकोआगुलेंट इज प्रेजेंट. राइट इन द कमेंट बॉक्स ये हमारी बॉडी में क्यों नहीं क्लॉट करता है. लेट्स कम बैक अगेन. मस्कीटो ने बाइट करा आपको, 2000 स्पोरोजोइट आपकी बॉडी में एंटर करा. आधे घंटे में इट इज गोइंग टू रीच टू द लिवर सेल. लिवर सेल में इट अंडरगोज टू फेजेस, प्री एरिथ्रोसाइटिक साइकिल एंड एक्सो एरिथ्रोसाइटिक साइकिल. लेट्स स्टार्ट विद द प्री एरिथ्रोसाइटिक साइकिल. सो स्पोरोजोइट हैज रीच टू द लिवर सेल. लिवर सेल में स्पोरोजोइट इज ट्रांसफॉर्म इनटू ट्रोफोजोइट. ट्रोफोजोइट हमने क्या पढ़ा था? इट इज द फीडिंग स्टेज. अगर वो खाएगा ही नहीं तो ग्रो कैसे करेगा? ग्रो नहीं करेगा तो रिप्रोडक्शन कैसे करेगा? रिप्रोडक्शन नहीं करेगा तो इन्फेक्ट कैसे करेगा? सो फर्स्टली इट हैज टू कन्वर्ट इनटू द फीडिंग स्टेज. सो स्पोरोजोइट कन्वर्टेड इनटू ट्रोफोजोइट. दीस ट्रोफोजोइट विल फीड. It will feed where? On the content of the liver cell, and then the sporozoite will feed on the liver cell and it will grow at its maximum size, or grow करके it will become spherical in shape. We call it as साइजोंट. इस सेल को हम साइजोंट बोलते हैं और इस स्टेज को हम साइजोंट स्टेज बोलते हैं. गेटिंग माई पॉइंट? नाउ इन साइजोंट न्यूक्लियस होता है. दिस न्यूक्लियस विल अंडरगो डिवीजन mitotically, okay, followed by cytoplasmic division. और ऐसा करके how many daughter cells are produced? 12,000 daughter cells are produced, which we call it as cryptozoite or first generation merozoite. Okay. One second. Okay, ठीक है. The cell membrane of cyzont burst. The entire process is completed approximately in 8 days. हमने टेबल में 8 days लिखा था. This releases the cryptozoite. It enters into liver sinusoid. Sinusoid is nothing but the minute blood vessels. For example, your capillaries which are present in the liver. Okay. Now the first generation merozoite or cryptozoite जो हमने पढ़ा, cryptozoite has two options. Either it is going to enter the fresh liver cell and continue the exoerythrocytic cycle, or it will enter into RBC and continue the erythrocytic cycle. Let's understand here. Sporozoite is going to enter where? In the liver cell, converted into trophozoite. Trophozoite will feed, grow at the maximum size. We call it as साइजोंट. Okay, यहाँ लिखा हुआ है साइजोंट. इस स्टेज को हम क्या बोलते हैं? साइजोंट stage. We call it as साइजोंट stage. Now इस stage के बाद what will happen? Nucleus will undergo mitotic division. Karyokinesis will occur first, followed by the cytokinesis, which results into 12,000 daughter cell. उस daughter cell का नाम अलग है, that is cryptozoite or first generation merozoite. Now ये क्या होगा? Cyzont cell is going to burst now. After bursting, cryptozoite will move out and it has two options. Either it is going to go and infect the fresh liver cell. अगर तो fresh liver cell को infect करता है तो we call it as it is going to follow exoerythrocytic cycle. अगर तो it is going to enter into RBC and infect the RBC, we call it as erythrocytic cycle. Okay. So this was about the pre-erythrocytic cycle जो पहले होता है before infecting the RBC. Now exoerythrocytic cycle अभी हमने देखा कि it has two option. तो अगर तो वो fresh liver को infect कर रहा है, कौन कर रहा है? Cryptozoite. If it is going to infect the fresh liver cell, we call it as exoerythrocytic cycle. Let's see here. If the cryptozoite enter into fresh liver cell, same process will take place what had happened in the pre-erythrocytic cycle. The only difference is that जो daughter cell बनेगा, it is going to be metacryptozoite. The daughter cell, हमने पहले एरिथ्रोसाइटिक साइकिल का जो डॉटर सेल बना उसको क्या बोला हमने? क्रिप्टोजोइट बोला था, राइट? यहाँ पे जो डॉटर सेल बन रहा है उसको हम क्या बोलेंगे? मेटाक्रिप्टोजोइट बोलेंगे. These metacryptozoites are of two type. One is smaller, other one is larger. Smaller one we call it as micrometacryptozoite. Larger one we call it as macrometacryptozoite. smaller one is going to infect the RBC. ऐसे याद रखो RBC छोटा सा होता है ना, तो it is going to infect RBC, and macrometa cryptozoite is going to infect the fresh liver cells. Same process. अगर तो वो RBC को infect करता है तो it is going to continue the erythrocytic cycle. अगर तो वो fresh liver cell को करता है तो it is going to continue the exoerythrocytic cycle, and this whole process takes place 2 days में. So exoerythrocytic cycle का कितना है? 2 days. 2 days, right? Now यहाँ पे आप देख सकते हो, यहाँ मैंने गलत लिखा है. What it is? Cryptozoite. Cryptozoite, okay? Cryptozoite is going to enter into fresh liver cell, and it is going to infect the fresh liver cell. Same process. Cryptozoite is going to turn into trophozoite, schizont, multiple karyokinesis, cytokinesis. The only difference is daughter cell जो बना है वो है metacryptozoite, then schizont burst. Metacryptozoite is released out. One is small, one other one is big. Smaller one we call it as micrometacryptozoite. Larger one we call it as macrometacryptozoite. Smaller one को हम क्या बोलते हैं? Micro. Larger one को हम क्या बोलते हैं? Macro. Now smaller one is going to enter into RBC. That cycle we call it as erythrocytic cycle. Larger one is going to enter into liver cells. That we call it as exoerythrocytic cycle. Right? Now let's enter into erythrocytic cycle. I want full attention over here. This is also called as Golgi cycle because it was described by Camillo Golgi. Now this cycle is initiated either by cryptozoite. You remember हमने सबसे पहला पढ़ा था pre-erythrocytic. वहाँ पे जो daughter cell बना था क्या बना था? Cryptozoite और first generation Merozoite, right? और हमने एरिथ्रोसाइटिक साइकिल में डॉटर सेल जो बनाया था वो क्या बनाया था? माइक्रो एंड मेटा बनाया था, जिसमें माइक्रो इज गोइंग टू इन्फेक्ट द आरबीसी. सो व्हाट आर द टू इन्फेक्टेड स्टेज फॉर एरिथ्रोसाइटिक? दैट इज क्रिप्टोजोइट एंड माइक्रोमेटा क्रिप्टोजोइट. सी हियर क्रिप्टोजोइट और माइक्रोमेटा क्रिप्टोजोइट इज गोइंग टू एंटर इनटू आरबीसी. आरबीसी में एंटर करने के बाद व्हाट इट इज गोइंग टू डू? दिस स्टेज assumes spherical shape and transform into trophozoite. So these two are going to convert into trophozoite. Why? Because trophozoite is the feeding stage. Feeding करेगा तभी तो grow करेगा तभी तो reproduce करेगा, right? Now these trophozoite, in trophozoite a vacuole appears. So suppose if this is the trophozoite, so vacuole appears which gradually grew in size, and it is going to push the nucleus and cytoplasm in one corner, that we call it as periphery. Now if you'll see the structure, it looks like a ring. तो इसको हम क्या बोलते हैं? Signet ring stage. Now after that it is going to lose the vacuole and it is going to form pseudopodia. So since pseudopodia is formed, what do we call it as? We call it as amoeboid stage. Now this amoeboid stage feed on the content of RBC. Amoeboid, याद है pseudopodia क्या करता था? It is going to engulf the food, right? So it is going to feed on the content of RBC, increase its size. RBC grows almost double in size. This growth, which usually double हो जाती है, this type of growth को हम बोलते हैं hypertrophy. This type of growth we call it as hypertrophy. Now here we have seen that it is going to eat the content of RBC. What is that content which is going to be eaten? It is nothing but your hemoglobin. Okay, hemoglobin. Now in this hemoglobin it is going to digest the whole globin part. The whole globin part is going to be digested. When we talk about the heme is nothing but the soluble substance. This heme is going to get converted into all insoluble crystalline substance, which we call it as hemozoin. So what happens to heme? Heme is a soluble substance which is converted into insoluble crystalline substance. We call it as hemozoin. Now this hemozoin we call it as malarial pigment. क्यों? क्योंकि जो हमने पढ़ा था ना tertian and quatern, that every third day fever occur, every fourth day fever occur, it is because of the hemozoin. Put star mark over here. It is the neat question. Why do we have fever? Whenever there is release of hemozoin, whenever RBC burst and hemozoin is released, there will be fever. Okay, fever कौन cause करता है? It is hemozoin. Okay, it is hemozoin. Now during this stage, small red color dot appear in the cytoplasm of the RBC known as Schaffner's dot. Now this Plasmodium vivax, obviously it is the parasite, it is the pathogen. So pathogen है तो antigen भी होगा, right? Pathogen है तो antigen भी होगा. What is antigen? Antigen is nothing but a foreign substance. Basically ये protein होते हैं, ठीक है? तो उनके टुकड़े, their this kind of, okay, इसको ऐसे बनाते हैं. Here you will be have Schaffner's dot. These dots are nothing but the antigen of Plasmodium vivax. Now Plasmodium loses the pseudopodia. So पहले it loses the vacuole, then it loses the pseudopodia and further increase in the size and occupies the entire RBC. हमने क्या पढ़ा था, अगर entire मतलब अगर बहुत ज़्यादा size में बढ़ जाता है तो what do we call it as? We call it as sizont. Now, sizont undergoes sizogony. कौन से sizogony? Erythrocytic sizogony, where it produces 12 to 24 daughter cells. And what is the name of daughter cell? Erythrocytic merozoite. हमने pre-erythrocytic में daughter cell क्या पढ़ा था? Cryptozoite. Okay. Exoerythrocytic में क्या पढ़ा था? Metacryptozoite. Right? And यहाँ पे erythrocytic cycle में हम क्या पढ़ रहे हैं? Erythrocytic merozoite. Now they are arranged, all these 12 to 24 daughter cells are arranged like this, which is nothing but like a petal of rose, So due to its appearance, we have given the name rosette stage, okay, or rosette stage. Finally, RBC burst. When RBC bursts, what comes out? Hemozoin comes out. This release of hemozoin and also erythrocytic merozoite will come out, right? Now, this cycle gets completed in 48 hours. That means your 2 days. 2 days में आपकी erythrocytic cycle complete हो जाती है. See this diagram over here. Cryptozoite or micrometacryptozoite is going to enter into RBC. It is going to convert into the trophozoite. Trophozoite mein there will be the formation of vacuole and which gradually increases and pushes the nucleus and cytoplasm in one corner, which gives a ring-like structure. So we have given the name signet ring stage. Now after that it loses the vacuole and there will be the formation of pseudopodia. Now since the formation of pseudopodia is there, हम इसको क्या बोलते हैं? Amoeboid stage बोलते हैं, right? Now sizont, after that what will happen? It is going to feed on the RBC. RBC पे feed करेगा कौन से substance पे? Hemoglobin पे. Globin it is going to engulf all. Heme क्या होता है? Heme is going to convert into insoluble crystalline, which we call it as hemozoin. After that it is going to grow in size. We call it as sizont. Sizont जब grow कर जाता है तो क्या हो जाता है? Karyokinesis होती है उसकी nucleus की, same होगा यहाँ पे. Then cytokinesis होती है, due to which there will be the formation of daughter cell. उनका नाम क्या है? Erythrocytic merozoite. Now these erythrocytic merozoites are arranged like a petal of rose, so we call it as rosette stage. Then RBC burst. When it bursts, erythrocytic merozoites released out and hemozoin is also released out. Now these hemozoins are pyrogen substance. Pyro means fire. तो क्या होता है? It's going to cause fever. Right? Now, so this was all about the one part of, that is your sizogony is over. Sizogony is over in human. The second part is nothing but gametocyte. We have seen that it will start the formation of gamete in human, right? After repeated cycle of erythrocytic sizogony, the fresh RBC decreases. Some merozoite enter the RBC and transform into gametocytes instead of continuing the erythrocytic cycle. So what will happen? एक तो हमारे पास ऑप्शन था कि मीरोजोइट इज गोइंग टू एंटर. कौन से मीरोजोइट क्लास हमने देखे थे? क्रिप्टोजोइट, दैट इज योर फर्स्ट जनरेशन मीरोजोइट, एंड देन योर माइक्रोमेटा क्रिप्टोजोइट, that is your second generation. Okay, these all are going to enter into RBC, right? So it has two options. One is nothing but after the repeated, one is nothing but what I was about to say, class. Okay, so one way is it is going to enter into the fresh RBC and infect the RBC. It is going to infect the RBC. Right? Other way is without infecting, it is going to enter and follow the gametocyte process. Okay. Now, along with, ठीक है, इतना काफी है. Now, कहाँ थे हम? This process generally takes place when the RBC are present in spleen and bone marrow. So it is going to enter and infect the fresh RBC apart from spleen and bone marrow, but mostly when they are in spleen and bone marrow, it is going to form gametocytes. Okay. Now these gametocytes can be smaller or larger. Smaller one we call it as microgametocyte. Larger one we call it as macrogametocytes. Right? Now, sorry, I forgot to say, smaller one is the male gametocyte and the larger one is your female gametocyte. The gametocyte cannot undergo further development in human. Why? Because of the temperature and pH of the blood. The what is required for gametocyte? What is gametocyte class? It is not a gamete. It is the gamete producing stage. Okay, it is going to produce male and female gamete, but it itself is not a gamete, right? So in mosquito, it will have proper temperature and pH. So there will be the formation of gametes. But here in human, only gametocyte start hota hai, but there won't be any formation of the gamete. These gametocytes reach blood circulation and wait to reach the next host. कैसे वो next host के पास जाएगा? Through mosquito. Through mosquito. First it is going to enter into the mosquito, right? They degenerated or die if not transferred to the mosquito within the week. तो क्या होता है? Gametocytes are formed. Then mosquito has to come and bite. When it sucks the blood, these gametocytes are going to enter into the mosquito. Suppose if the mosquito doesn't come and bite you, so what will happen? These Merozoites are going to be not Merozoites, sorry. These gametocytes are going to get degenerated in your body. Correct? Now, the another topic is pre-patent period and incubation period. What is incubation period, class? It is nothing but sporozoite, which is the infective stage. From infective stage till the symptom, we call it as incubation period. So from sporozoite till symptoms, we call it as incubation period. Incubation period क्या होता है, class? It is nothing but जब तक आपको symptoms दिखना शुरू नहीं हो जाते. और symptoms कब दिखते हैं? जब वो disease already आपकी body में grow कर चुका है. Okay, grow कर चुका है. Then only you will have the appearance of symptoms. Entry करते ही symptoms नहीं होते हैं, right? So वो period of time जब तक वो symptoms दिखाता है, तब तक को हम क्या बोलते हैं? Incubation period. What is pre-patent period? It is nothing but sporozoite to cryptozoite. We call it as pre-patent period. Now everything I have written in table. Pre-patent is nothing but sporozoite to किधर गया? Cryptozoite. We call it as pre-patent period. Incubation is nothing but sporozoite to symptoms. We call it as incubation period. Duration, it is for 8 days and this one is for 10 to 14 days. Only multiplication occur in pre-patent period. क्योंकि सिर्फ multiplication होती है, we won't be able to see the symptoms. But यहाँ multiplication तो होती है, but only multiplication होती है क्या? No. Multiplication होती है and then harming the host cell होती है, due to which what will happen? The symptoms will occur, right? So this cycle you can see here, class. First one is pre-erythrocytic cycle. That is nothing but sporozoite enters the liver cell, which converted into trophozoite. Trophozoite converted into cyzont. Then erythrocytic ये कैसे होता है? साइजोगोनी हो रही है. कौन सी साइजोगोनी हो रही है? Hepatic साइजोगोनी. Hepatic साइजोगोनी, right? Now, then daughter cells are formed, which we call it as cryptozoite. Now, cryptozoite, what will happen after that? Cryptozoite has two options. Either it will enter into RBC or it is going to infect the fresh liver. So if it infects the fresh liver, again क्या होता है? Cryptozoite is converted into trophozoite. Cyzone stage होती है. Daughter cells are produced. इस daughter cell को हम क्या बोलते हैं? Micrometa cryptozoite. अगर तो it is going to enter into RBC, it is going to again convert into trophozoite. Trophozoite के बाद vacuole formation होती है, which is going to push nucleus and cytoplasm in one corner, which stage we call it as signet ring stage. Then vacuole loose हो जाता है. There will be the formation of pseudopodia. We call it as amoeboid stage. Then यहाँ पे cyzone, it grows, it feeds on the hemoglobin. Globin part completely destroy कर देता है heme में. It converted into insoluble hemozoin, right? And there you will see antigen of the Plasmodium vivax. We call it as Schaffner's granules. Then there will be the formation of hemozoin. अभी बता दिया मैंने आपको. Then daughter cells are formed. These daughter cells are erythrocytic merozoite. This daughter cells are erythrocytic merozoite, and these merozoite, when the cell bursts, it is going to infect the किधर लिखा? Erythrocytic merozoite enter an RBC. The fresh RBC, it is either going to enter into the fresh RBC or it is going to enter into the another RBC. Fresh RBC only. It is going to enter कहाँ पे? Spleen में and bone marrow की RBC में. Bone marrow की RBC में, and it is going to form gametocyte, male gametocyte, and where is the female? Female gametocyte. Okay. Now class, let me correct one thing over here. Here class, I have written this merozoite. Now this is not cryptozoite and micrometa cryptozoite. Which merozoite we are talking about? We are talking about the erythrocytic merozoite. Erythrocytic merozoite. As per your diagram, it is erythrocytic merozoite. So let me explain this again. So there will be the formation of erythrocytic zygogony. Kaise? By erythrocytic, sorry, there will be the formation of erythrocytic merozoite by the end of erythrocytic zygogony. These erythrocytic merozoite has two options. It will enter into the fresh RBC and go infect the RBC. Let me correct it over here. So, one second, let me run this. Okay, not this one. Okay, which one is going to enter? Then, erythrocytic merozoite. It is going to enter into fresh RBC. या तो fresh RBC में infect करेगा और RBC of spleen or which one? Bone marrow. Spleen or bone marrow. It is going to form gametocyte. That is going to be male gametocyte and female gametocyte. Right? So that was all about the Plasmodium life cycle in men. Now let's see the Plasmodium life cycle in female Anopheles mosquito. Female Anopheles, so whole that cycle has occurred in the male. Now अब क्या होता है? Female Anopheles mosquito will come and bite the malarial patient. Malarial patient क्यों बोल रहे हैं? Because hypnozoin is released, right? Now सब कुछ वहाँ पे हो चुका है. This cycle we also call it as Rose cycle. This cycle we also call it as Rose cycle because Rose was the scientist who found this, right? Now mosquito is going to come, bite the malarial patient and suck the blood. Blood suck करा तो उसमें gametocytes and all the other stages, your cryptozoite, microcryptozoite, all the other stages जो भी blood में present है, all the other stages are going to enter inside the mosquito. But all these stages are digested except your gametocytes. Okay, only gametocyte will survive. Rest all the other stages. Why? Because उसकी body में that kind of environment is not present where all the other stages will stay alive. Right? Only gametocyte will stay alive. Now these gametocytes undergo four process over here. One is your gametogony, second is your fertilization, third is your formation of ookinete or oocyst, and fourth one is your sporogony. Now these gametocytes, what are gametocytes? Gamete forming, okay, gamete forming substance. It is not going to form your, let me show you, I have written somewhere now. Gamete producing stage, we call it as gametocyte. It is not the gamete, it is not the gamete, right? This is resistant to the digestive enzyme. Now, gametogony class, what is gametogony? Let's start with gametogony. It is nothing but gametogony, formation of gamete. कौन सा gamete? Male and female gamete. Formation of male and female gamete, we call it as gametogony. Where it occurs? It occurs in the lumen. पूरा ह्यूमन में जो साइकिल हुआ था वो हमने कहाँ देखा? लिवर सेल में देखा एंड वी हैव सीन इन द आरबीसी, राइट? लिवर सेल एंड इन द आरबीसी. यहाँ पे कहाँ हो रहा है? मस्किटो की ल्यूमेन. किसकी ल्यूमेन में? क्रॉप में. व्हाट इज क्रॉप? क्रॉप इज नथिंग बट योर डाइजेस्टिव सिस्टम का पार्ट है. इट इज द पार्ट ऑफ द डाइजेस्टिव सिस्टम. इट इज द पार्ट ऑफ द डाइजेस्टिव सिस्टम. जैसे हमारा स्टमक होता है उनका क्रॉप होता है. ओके, नाउ क्रॉप का ल्यूमेन. व्हाट इज ल्यूमेन? द एम्प्टी स्पेस विच इज प्रेजेंट इनसाइड. द एम्प्टी स्पेस विच इज प्रेजेंट इनसाइड वी कॉल it as lumen. The empty space which is present inside. तो ये process gametogony, formation of male and female gamete कहाँ हो रही है? क्रॉप के empty space में हो रही है. Right? Now see here. In microgametocyte, हम male and female का पढ़ रहे हैं. So first we will study about the male. In microgametocyte nucleus, it undergoes three mitotic division. It undergoes three mitotic division. Before that, let's just revise one thing class. We have already studied. अगर हम male और female के gamete के बात करते हैं, तो this gamete का nucleus को हम क्या बोलते हैं? Pronuclei बोलते हैं. We have given the special name to the nucleus of the gamete that we call it as pronuclei. So in microgamete, थ्री माइटोटिक डिवीजन टेक्स प्लेस. थ्री माइटोटिक डिवीजन मतलब यहाँ पे एक डिवीजन किया तो कितना न्यूक्लियाई हुआ? 2 बना. फिर दूसरा करा तो 4 बना. फिर तीसरा करा तो 1 2 3 4 5 6 7 8. हाउ मेनी? 8 न्यूक्लियस विल बी फॉर्म. हाउ मेनी? 8 प्रोन्यूक्लियाई विल बी फॉर्म, राइट? नाउ दीज 8 प्रोन्यूक्लियाई विल रीचेस टू द पेरिफेरी. ये पेरिफेरी पे अरेंज हो जाते हैं. एंड नाउ साइटोप्लाज्म इज पुश्ड. यहाँ से साइटोप्लाज्म बाहर की तरफ पुश करता है. ड्यू टू विच व्हाट विल हैपन? लेट मी शो यू ओवर हियर. इधर देखो. सो यहाँ तक हमने पढ़ लिया था. पेरिफेरी पे आ गया है. देन साइटोप्लाज्म इज पुश्ड outward due to which इस तरीके का structure, wave like structure is formed. Wave like structure is formed, right? Now see here. किधर गया? हाँ, साइटोप्लाज्म इज पुश्ड आउटवर्ड इन द फॉर्म ऑफ 8 फ्लैजिला. सो दिस वेव लाइक स्ट्रक्चर जो हमने देखा था ना, यही एक्सटेंड होके क्या बन जा रहा है, फ्लैजिला बन जा रहा है. इट इज गोइंग टू फॉर्म फ्लैजिला. ईच फ्लैजिला लाइक प्रोसेस यहाँ इज नथिंग बट योर आउटग्रोथ. प्रोसेस इज नथिंग बट योर आउटग्रोथ. रिसीव्स वन प्रोन्यूक्लियाई. अब जो न्यूक्लियाई यहाँ पे थे ना, इट इज गोइंग टू एंटर इनसाइड दिस फ्लैजिला लाइक आउटग्रोथ, फॉर्म माइक्रोगैमेट्स. और ये क्या हो जाता है, these are going to form 8 microgametes. यहाँ देखो, ये जो माइक्रोगैमेट्स है, इट शोज द लैशिंग मूवमेंट. इस तरीके से साइडवाइज लैशिंग मूवमेंट शो करता है, जिसकी वजह से इट गेट्स सेपरेटेड फ्रॉम द साइटोप्लाज्म. एंड क्या इस प्रोसेस को, जहां सेपरेशन ऑफ दिस फ्लैजिला, ये 8 फ्लैजिला जो बना है, फ्लैजिला लाइक प्रोसेस जो बना है, इट इज गोइंग टू गेट सेपरेटेड फ्रॉम द साइटोप्लाज्म. उस प्रोसेस को हम क्या बोलते हैं, एक्सफ्लैजिलेशन बोलते हैं. एंड फाइनली 8 माइक्रोगैमेट्स आर फॉर्म. इधर देखो, यहां तक तो समझ आ गया था. नाउ इस साइटोप्लाज्म को आउटर तरफ पुश किया, जिसकी वजह से फ्लैजिला लाइक प्रोसेस बना. ठीक है, इस फ्लैजिला लाइक प्रोसेस में क्या होता है, तुम्हारा एक एक प्रोन्यूक्लियाई इज गोइंग टू एंटर ओवर हियर. देन एक्सफ्लैजिलेशन ऑकर. एक्सफ्लैजिलेशन मतलब क्या होता है, दिस फ्लैजिला, ये मान लो इस एक फ्लैजिला को ले लेते हैं, इट शोज साइडवाइज मूवमेंट. इस तरीके का मूवमेंट शो करता है, ड्यू टू विच इट गेट डिटैच फ्रॉम द साइटोप्लाज्म एंड इट इज गोइंग टू फॉर्म 8 गैमेट. कौन सा गैमेट, मेल गैमेट, राइट. नाउ फीमेल गैमेट में कुछ भी नहीं है. माइक्रोगैमेटोसाइट अंडरगो मैच्योरेशन टू ट्रांसफॉर्म इनटू फीमेल गैमेट. न्यूक्लियस ऑफ फीमेल गैमेट मूव टुवर्ड्स द पेरिफेरी. जैसे वहां पे पेरिफेरी में मूव किया था मेल में, यहाँ पे भी इट इज गोइंग टू मूव टुवर्ड्स द पेरिफेरी. एंड साइटोप्लाज्म एट द पॉइंट फॉर्म द प्रोजेक्शन. सो यहाँ पे प्रोजेक्शन फॉर्म होता है एंड दिस वी कॉल इट एज फर्टिलाइजेशन कोन. यहाँ पे देखो, यहाँ पे क्या हुआ, यहाँ तक समझ आया ना, मैच्योरेशन हो गया. आफ्टर दैट प्रोन्यूक्लियर मूव टुवर्ड्स वन कॉर्नर एंड साइटोप्लाज्म, जहाँ पे वो कॉर्नर में मूव किया है, साइटोप्लाज्म इज गोइंग टू प्रोजेक्ट आउट टू फॉर्म द फर्टिलाइजेशन कोन, राइट. न्यूक्लियस आल्सो मूव टुवर्ड्स द पेरिफेरी एंड दिस फर्टिलाइजेशन कोन, नाम ही क्यों दिया फर्टिलाइजेशन कोन, दैट मींस फर्टिलाइजेशन टेक्स प्लेस ओवर हियर, राइट. वो पूरा प्रोसेस हमने कहाँ देखा, ल्यूमेन में देखा. देन देयर इज फर्टिलाइजेशन. व्हाट इज फर्टिलाइजेशन, फ्यूजन ऑफ गैमेट्स बोलने से रॉन्ग हो जाएगा. फ्यूजन ऑफ मेल एंड फीमेल गैमेट्स वी कॉल इट एज फर्टिलाइजेशन. ये भी कहाँ होता है, इट ऑकर्स इन द ल्यूमेन ऑफ द क्रॉप. what this process involves, it involves the formation of the zygote. It involves the formation of the zygote. See here, male gamete enter into female gamete through fertilization cone. So यहाँ पे हम बना देते हैं ये वाला बनाते हैं. This is your अरे सेम बना दिया. Okay, ठीक है, let it be. Fertilization cone. So male is going to enter over here. Male का sperm जो है, this is male sperm. It is going to enter over here. कहाँ से, through fertilization cone. So male gamete is going to enter female gamete through fertilization cone. Both male and female gamete fuse together. तो fusion में सबसे पहले fuse क्या करता है, that pronuclei will fuse, right? Pronuclei fuse together. This process of fusion of pronuclei we call it as एम्फीमाइसिस. ओके, एम्फीमाइसिस वी कॉल इट एज द प्रोसेस जिस प्रोसेस से प्रोन्यूक्लियर फ्यूज करते हैं, एम्फीमाइसिस बोलते हैं. बट फ्यूज होने के बाद द न्यूक्लियस व्हिच इज फॉर्म, उसको हम क्या बोलते हैं, सिनकैरियन बोलते हैं. वी हैव ऑलरेडी स्टडीड दिस इन लोकोमोशन एंड रिप्रोडक्शन, राइट. फ्यूज होने के बाद जो बनता है, वी कॉल इट एज सिनकैरियन. एंड फ्यूजन ऑफ मेल एंड फीमेल गैमेट के बाद जो सेल बनता है, वी कॉल इट एज जाइगोट, व्हिच इज राउंड एंड नॉन मोटाइल इन केस ऑफ प्लास्मोडियम वाइवेक्स. नाउ यहाँ पे अगर हम देखेंगे क्लास, तो मेल वन इज स्मॉलर, माइक्रो बोला था ना, मेल वन इज स्मॉलर एंड फीमेल वन इज लार्जर, दैट इज मैक्रो. Micro and macro female fuse together. So are they similar or dissimilar? They are dissimilar gametes. So fusion of dissimilar gamete, what do we call it as? We call it as anisogamy. We call it as anisogamy. Then third step is formation of ookinete or oocyst. अब देखो, zygote was round and non-motile, so it will remain inactive for some time. After 18 to 24 hours, it transform. This round zygote is going to transform into long, slender, vermiform. मतलब long only, ठीक है, thread-like. It is going to form long, slender structure. Now this structure we call it as ookinete. What is ookinete? Nothing but the zygote जो round था, it got converted into long, slender like structure which we call it as ookinete or vermicule. Now ookinete pierce the wall of crop. अब तक सब कुछ कहाँ हो रहा था, ल्यूमिन में हो रहा था, यहाँ हो रहा था. Now this vermiform is going to pierce the wall of क्रॉप का वॉल को पियर्स मतलब होल करना. इट इज गोइंग टू पियर्स द वॉल ऑफ क्रॉप एंड सेटल बीनीथ द बेसमेंट मेम्ब्रेन. क्लास हमेशा एपिथेलियल मेम्ब्रेन जो होती है, एपिथेलियल टिश्यू जो होता है, उसके नीचे ऑलवेज देयर विल बी द प्रेजेंस ऑफ बेसमेंट मेम्ब्रेन फॉर नरिशमेंट. ओके, ऑलवेज देयर फॉर नरिशमेंट एंड सपोर्ट. ऑलवेज बिलो द एपिथेलियल मेम्ब्रेन देयर विल बी द प्रेजेंस ऑफ बेसमेंट मेम्ब्रेन. सो यहाँ भी एपिथेलियल मेम्ब्रेन के नीचे देयर विल बी द प्रेजेंस ऑफ बेसमेंट मेम्ब्रेन. तो यहाँ पे देखो, इट इज गोइंग टू settle beneath the basement membrane, right? Now this ookinete is going to become round in structure. Again round structure ban jayega and it is going to secrete cyst around itself. It is going to secrete cyst around itself. Now we call it as oocyst. कुछ भी नहीं है class. Zygote tha which was round in shape. This round in shape got converted into long cylinder like structure. We call it as ookinete. This ookinete is going to pierce the wall and enter beneath the basement membrane. वहाँ पे वो क्या करेगा? It is going to again change its structure. It becomes round from long and slender and it is going to develop cyst around itself. इस structure को हम क्या बोल देते हैं? oocyst बोल देते हैं, right? Now 50 to 500 oocysts are formed on the wall of the crop and appears in the form ऑफ द नोड्यूल. इस तरीके से फॉर्म होते हैं क्लास, नोड्यूल बन जाते हैं. ठीक है, नोड्यूल विल बी देयर. यहाँ पे आप डायग्राम में देख सकते हैं फर्टिलाइजेशन कौन है. इट इज गोइंग टू फर्टिलाइज थ्रू हियर एंड आइसोगेम इस प्रोसेस को बोलते हैं. फर्टिलाइजेशन होती है, जाइगोट बनता है विच इज राउंड, नॉन मोटाइल एंड इनएक्टिव फॉर सम टाइम. आफ्टर 18 to 24 hours it changes its shape into long cylinder like structure. We call it as ookinete. Ookinete is motile, so it is going to pierce the wall, enter beneath the basement membrane and around it it is going to develop the cyst. Now we call it as oocyst. This oocyst, 50 to 500 oocysts will be there and it appears like a nodule. What is sporogamy? It is nothing but the formation of sporozoite. According to Gano, the nucleus of oocyst. अभी तक हमने कोई division नहीं देखी थी, just fertilization देखा and change in the shape देखा. अब oocyst का nucleus, already cyst है ना यहाँ पे. Okay, now nucleus is undergoing reduction division. That means first meiosis will occur, okay, followed by the mitosis division, results into 1000 daughter nuclei. This nuclei को हम क्या बोलते हैं? इधर लिखा है, wait one second. Okay, we'll read, let it be. Okay, so now this 1000 daughter nuclei is formed, around which what will happen? Cytoplasm will surround it and this is going to transform into sickle cell sporozoite. Okay, देखो पूरा बता रहे हैं क्या होता है. यहाँ पे आपका मेल, एक सेकेंड, मेल है and female. They are going to fuse together through the process of fertilization and we call it as anisogamy. It is going to form round zygote. This round zygote is non-motile. Okay, now this round zygote is going to convert into slender long structure. We call it as ookinete. कहाँ पे हो रहा है? अब तक सब lumen में हो रहा है. ठीक है, now this ookinete is going to enter into, it is motile, it is going to enter beneath the basement membrane and again convert into round structure and it has cyst around itself. Now we call it as oocyst. Now this oocyst, what will happen? It undergoes karyokinesis. First meiosis division will occur, meiosis followed by the mitosis and which results into the daughter cells, 1000 daughter cells. Now these daughter cells are going to transform into sickle shaped structure. We call it as sporozoite. Transform into sporozoite, right? Now the oocyst where the sporozoite, ये sporozoite कहाँ बना है class? ये तो पूरा oocyst है ना, इसके अंदर sporozoite बना है. So when oocyst is having sporozoite, उस stage को हम क्या बोलते हैं? Sporocyst बोलते हैं. What do we call it as? Sporocyst. So जैसे ये sporocyst rupture करता है, what is going to come out? 10,000 sporozoites are going to come out and liberate into the hemocoel. Seal क्या होता है? It is a cavity. It is a cavity. Now hemocoel है ना, तो in this cavity what is going to be present? Hemolymph is present. What is hemolymph? Hemolymph is nothing but invertebrate में instead of blood you are going to see what do you call, hemolymph. In invertebrate, instead of blood, you are going to see hemolymph. So it is going to transport the nutrient, excretory material and all these things. Okay. Now these sporozoite is going to migrate into salivary gland and they are ready for the infection. जैसे ही Plasmodium mosquito will bite a human, this sporozoite is going to enter inside. Now this whole the life cycle of Plasmodium and mosquito is completed into 10 to 24 days. This whole 4 stage is going to be completed in 10 to 24 days. यहाँ पे देखो oocyst फॉर्म किया, meiosis हुआ, repeated mitosis हुआ, karyokinesis हुई, cytokinesis हुई and it's got converted into sporozoite. Then oocyst rupture होने sporocyst rupture and rupture होने के बाद there will be the release of what? Release of sporozoite. This sporozoite goes to salivary gland of the mosquito. Through what? Through hemocoel, right? So here you can see class, mega gametocyte form mega gamete. Okay, that means female gamete. Then micro will form male gamete. So यहाँ पे fertilization हो रही है आपकी male and female gamete fertilization cone में. Then ookinete form हो रहा है and stomach wall आपकी दिख रही है यहाँ पे. क्या पढ़ा था? Stomach wall में infect करेगा, right? Then there will be the formation of cyst wall. उसको हमने oocyst बोला. Then nuclei will undergo karyokinesis followed by cytokinesis and this results into the formation of sporozoite. Then oocyst burst, that is sporocyst burst and it is going to liberate out sporozoite and this sporozoite will enter into the salivary gland of the mosquito through hemocoel, through hemolymph actually. Okay. पैथोजेनिसिटी देखे तो बेनाइन टर्शियन मलेरिया है. क्लिनिकल सिम्टम्स बाउट ऑफ फीवर होते हैं. दैट मीन्स फीवर विल ऑकर इन कैप आफ्टर एवरी 3rd डे और आफ्टर एवरी 4th डे. इनकी 3 स्टेजेस होते हैं, दैट इज कोल्ड स्टेज, हॉट स्टेज और स्वेटिंग स्टेज. कोल्ड स्टेज में क्या होती है आपका चिल एंड शिवरिंग होती है. व्हाई? ड्यू टू रप्चर ऑफ आरबीसी. हॉट स्टेज मीन्स ऑब्वियसली हाई टेम्परेचर होगा, दैट इज 106 फ़ारेनहाइट. क्यों होता है? ड्यू टू रिलीज ऑफ हीमोजोइन. ड्यू टू रिलीज ऑफ हीमोजोइन. स्वेटिंग मीन्स abundant huge amount of sweating will occur to lower down the temperature. हमारी बॉडी का टेम्परेचर इतना हाई है कि my body wants to get it down. Now it causes anemia and splenomegaly. Anemia is nothing but deficiency of RBC or hemoglobin. And splenomegaly, spleno means spleen की बात करें, megaly means abnormal enlargement of the organ. Okay, abnormal increase in the size को हम megaly बोलते हैं. So there will be abnormal enlargement of the spleen, we call it as splenomegaly. Now chronic malaria में आप देखेंगे prolonged form of the malaria where infection persists for the extended form, बहुत ज़्यादा period के लिए malaria रहता है. Now relapse, that means वापस से reappearance of the malaria may be can be seen in some of the patient. Now why spleen enlarges in the case of chronic malaria? Due to repeated infection, and abnormal. See, spleen is also your lymphoid organ. It is the lymphoid organ. So basically it is used in the immunity also. It is used in the immune system or immunity also, right? Now relapse, that means reoccurrence of malaria. Now sometimes what happen, the macro metacryptozoite, that means जो liver को infect कर